Hello everyone, welcome to the 5th unit of AP Bio Review session. Now 5th unit is about meiosis and Mendelian genetics. Now let's first talk about meiosis. Before we talk about meiosis, we have to know what mitosis is, which I explained in the previous video. Now mitosis and meiosis are very similar, but they're slightly different, and I'll explain how they're different, okay? Now first of all, mitosis. The main purpose of mitosis is cell division, cell replication, one cell dividing into two. Now, why is this necessary? Of course, humans need to grow, or not just humans, but any organisms need to grow. And in order to grow, we need more number of cells, right? And how we get more cells is basically one cell splits into two. So mitosis was all about one cell dividing into two. Now, meiosis is slightly different. Meiosis is when one cell splits, but not into just other normal cells, but into gametes or sex cells. So that is the purpose of meiosis, and that is where the difference comes in. Mitosis is replicating literally the same cell as myself, but into two, so splitting myself into two, whereas meiosis is when one cell splits into multiple cells, but into sex cells. Now, or gametes. Now the difference between sex cells and just normal cell is that in sex cells, instead of having 46 chromosomes, you only have 23 chromosomes. So now let me explain what this actually means. Now, you would have already heard that humans have 23 pairs of chromosomes, right? Yes, we do have 23 pair of chromosomes. Now, each pair has 2 chromosomes, so in total we would have 46 chromosomes, okay? However, this is a normal cell. A normal cell would have full 46 chromosomes, but in gametes or sex cells, you only have 23 of these. You only receive 1 out of 2 from each pair, ending up with 23, not pairs of chromosomes, but literally 23 individual of chromosomes. Now, you might ask me, so then why do sex cells only have half the number of of chromosomes? Well, the reason is because how sex cells work is one sex cell from your mom, one sex cell from your dad combined to make your full cell, right? So in order for your cell to have 46 chromosomes or 23 pairs of chromosomes, each sex cell, your mom's sex cell, should have 23 chromosomes, and your dad's sex cell should also have 23 chromosomes. So that when the two combine, you have 23 pairs, and in total, 46 chromosomes, okay? So that is the difference between gametes, sex cells, and just normal cells, okay? Now let me explain how meiosis happens. Now, if you think about mitosis, you had 2 big stages, right? You had interphase, and you had mitosis. Now, when it comes to meiosis, it's pretty much the same. You have interphase, and you have mitosis, meiosis, but unlike mitosis, meiosis happened through 2 steps. So you have meiosis 1 and you have meiosis 2. Now interphase looks pretty much the same as mitosis. You have G1 phase, you have S phase, you have G2 phase. Okay? So the same thing literally happens. In G1 phase, you have cell growth, just like mitosis. In S phase, sister chromatids will be made, right? So initially, chromosomes look like this when the cell is a baby, but once it hits puberty, once it reaches S phase, each chromosome will duplicate to make sister chromatid. Each strand of chromosome is called a chromatid as well. Now, if this chromatid duplicates and you have 2 chromatids sticking to each other, it's still 1 chromosome, right? Because it's 1 piece. But it's now called the 2 chromatids. Why? Because it has 2 strands, and each strand is called chromatid. Now, in the previous video, I already defined the terms, the difference between chromatid, chromosome, centriole, centromere, and everything. So if you don't know those terms very well, I advise you to watch the previous video. So now Again, in S phase, these sister chromatids will be made, and in G2 phase, cell growth happens so that the cell can actually be prepared for meiosis 1 and meiosis 2. Now let me explain what happens in meiosis 1. Now in mitosis, what happened was all of the duplicated cells, or chromosomes, will align in the middle, right? And they will rip off like this. Right? Now in meiosis 1, instead of all of the chromosomes lining up individually, each chromosome will find its partner and line up in the middle with its partner like this. Like this. Do you see the difference? Over here, individually they lined up in the middle, but here, each chromosome will find its partner and line up together. And once it splits, of course, individual full chromosome will be split apart instead of one chromosome being split into two pieces, right? How is this possible? Again, this is possible because two chromosomes lined up together. So mitosis is like this. I line up in the middle and my left part of the body and right part of the body gets ripped off. So I split into two pieces. Whereas in meiosis, step one, what happens is I line up in the middle with my partner. So my partner will be split out from me and I will be split to the other side. So yes, still splitting apart is happening, but instead of my body, instead of chromosome literally splitting into half, what's going to happen is one of the partner will be split to the left and the other partner will be split to the right. So that's the difference that we're seeing here, okay? So more specifically, how meiosis 1 looks like is we still have PMAD, right? So in prophase, again, you will just have these centrioles, centromeres, and spindle fibers starting to appear. In metaphase, what's going to happen is, again, these chromosomes will find their partners, and with their partner, they will align into the middle, like this. In anaphase, the actual splitting part will happen. But again, no chromosome is actually being splitted. Only the chromosomes are being split from their partner. No actual individual chromosome's body is being split into two. Now finally, in telophase, these splitted chromosomes will be allocated into two different membranes. Okay? So now, in meiosis 1, what you have to remember is initially, you had 4 chromosomes, right? But end result is you only have 2 chromosomes per new cell. So now, what we can say is if this was a simplification, and if we're talking about human body, we start with 46 chromosomes, but the end result is each cell will only end up having 23 chromosomes, which is again what we were looking for earlier, right? Because as I told you, in sex cells, there's only half the number of chromosomes. Why? Because again, 2 sex cells meet to make a baby cell, so each sex cell should have half the number of cells that an actual normal cell should have. That is why we only have half the number of chromosomes left. Now, if you look at mitosis, even after splitting, yes, we have less chromatids, we have less strands, but if you look at individual particles, we still have 4 particles, right? We have 1, 2, 3, 4 chromosomes, and here we still have 1, 2, 3, 4 chromosomes. Yes, each chromosome is smaller, but still we have the same number of chromosomes, right? If it was human, we start with 46 chromosomes and we end with 46 chromosomes. That is mitosis, because again, mitosis is all about making literally the same version of myself because we're replicating. That is why in mitosis, the number of chromosomes don't change, but in meiosis, the number of chromosomes will be halved. And that process, that changing of number of chromosomes is called diploid to haploid transformation. So initially, you have diploid, di, carbon dioxide, di always means 2. You had double the number of chromosomes, but that would change to half the number of chromosomes. Thus, we call that a haploid cell. So this end result cell will be called a haploid cell, whereas this starting cell will be called diploid cell. Now, once we have this, and once each cell looks something like this cell now goes through meiosis, and meiosis 2. And meiosis 2 is very similar to mitosis because meiosis 2 again would have PMAD, right? What happens in prophase is literally the same as mitosis. There will be centromere and spindle fibers coming out from it. In metaphase, the chromosomes, again, this time they don't find partners, but they individually align in the middle, just like mitosis, right? Over here, they found partners together, but over here, they're not finding partners. They're individually lining up in the middle, connected to spindle fibers. Now, in anaphase, what happens is, since individual chromosomes are aligned in the middle, the individual chromosomes will be split into two body parts and be ripped off like this. Do you understand? It's the exact same as mitosis. And again, in telophase, finally, you will have two end result cells made with same number of chromosomes as here, but half the number of chromosomes as initial cell, right? So that is why we call this a haploid cell again. So this was how meiosis 2 happens. And so the total end result is you start with a normal cell, right? And you will end up with 4 sex cells with only half the number of chromosomes in each of the cell. Now, one important thing to remember in this whole process is here. Now, when a chromosome finds its partner to be aligned in the middle, the partner and I will share some body parts. I'll cut my left leg, give it to my partner, and my partner will cut her left leg and give it to me. So we exchange some body parts, and that process is called crossover. Now, why is crossover necessary? Well, we don't want to pass down the exact same chromosomes to my baby, right? Of course, it would be great if my baby looks like me, but I don't want my baby to look exactly like me. That would be kind of creepy. We want to kind of make some diversity, and the process of making that diversity is changing a bit of my chromosome with my partner's chromosome and mixing the two so that, yes, I am sharing my genetics, but also I'm sharing my partner's genetics, and it's being mixed. Right? That is the whole point of the crossover. We want to increase diversity because, again, diversity is very beneficial when it comes to natural selection and surviving in different types of environments. So crossover, remember crossover. Another important thing is called random assortment. Now, random assortment is basically when these partners align in the middle, this order is random. Now, why is this important? If they always align in the middle in the exact same order, then how these 4 chromosomes split into 4 smaller sexes will literally be the same every single time. And again, that decreases diversity, right? So how these these partners align will always be different. It's going to be random all the time. And by that way, we can increase the diversity of this whole population. Okay, so that is it for meiosis, and now I'll be moving to Mendelian genetics. Now let's move on to the Mendelian genetics. As you can tell from the change in the background, I had to record this again. Why? Because my previous video had such a bad explanation in this part, Mendelian genetics. So I'm recording this one more time. So I'm gonna be better, hopefully. Now, Mendelian genetics, basically, each of your trait, whatever trait you're talking about, whether it's your hair color, whether your hair is straight or curly, your eye color, the shape of your eye, whatever, is always coded based on 2 alleles. One allele you will receive from your mom, and one allele you'll receive from your dad. Okay? By combining 2 alleles, 2 genetic information, one from your mom and one from your dad, you will decide which trait you will actually express in your body. For example, if your mom gives straight hair, if your dad also gives straight hair allele, then probably you will have straight hair on your body, right? However, apparently, Mendelian genetics tells us that this is not necessarily true, and I'll explain to you why. Now, first of all, we need to know some terms. First is genotype. Second is phenotype. Now, genotype is literally these codes, these combinations of alleles that you possess. It's the code that will make phenotype occur. And phenotype is the actual physical attribute that are observable. So in this case, genotype would be straight allele from your mom and straight hair allele from your dad, and the phenotype would be the actual straight hair you will be observing. Now in this case, you might think, wait, the genotype and phenotype are literally the same. They're both just straight hair, straight hair. Now, there are some exceptional cases. For example, let's say your mom has straight hair, your dad gives you curly hair genotype, sorry. So you have these 2 combination of codes, but your actual hair will end up in straight hair. Now what you're seeing over here is you're getting different codes, and these codes are what we call genotype, but the phenotype, the actual physical attribute that we're observing will be just straight hair. Now, then of course, when we're receiving two alleles, how do we decide, depending on the combination of two alleles, what type of physical attributes will we be observing? Well, that is what Mendelian genetics tries to observe. Now, first of all, we have to know two different types of alleles. In Mendelian genetics, there are only two different types of allele. One is recessive, the other one is dominant. As you can tell from the name, dominant allele is the allele that is much more likely to override, not much more likely, but it will always override the recessive allele. So, for example, in this case, between straight hair and curly hair, straight hair is a dominant trait and curly hair is a recessive trait. That is why, if you receive one straight hair allele and one curvy hair allele, then you will end up having straight hair phenotype. Why? Because straight hair is dominant trait, so straight hair allele will override this recessive allele, this curly allele, and end up making straight hair in your body. So this is the whole point of Mendelian genetics. Mendelian genetics tells us that all characteristics in our body are made out of two alleles, one from your mom and one from your dad. And depending on that combination of two alleles, you will either have a recessive trait, a dominant, or a dominant trait. And more specifically, there are two different types of dominant traits. One is homozygous dominant, and another is heterozygous dominant. Now, heterozygous dominance happens when, again, you get one dominant allele and one recessive allele. Still, since dominant allele overrides recessive allele, you will end up having dominant trait. But so the phenotype will just be will just seen as dominant characteristic, right? However, if you look at the genotype, you can see how there's a mixture in the two alleles, right? In this case, we call it a heterozygous dominant trait that you're observing. Now, there are some cases where you receive two dominant alleles. For example, straight hair allele and straight hair allele. In this case, yes, you will observe same physical attribute as the case above, which is straight hair, but the specific code that is actually making that straight hair appear in your body is different because they're both the dominant alleles. In this case, we call it homozygous dominant. Now, of course, homo means same, right? So homozygous dominant would have two same dominant alleles. That would be homozygous dominant trait, whereas heterozygous dominant trait is when you have one dominant allele and one recessive allele, and the two combine, and one overrides the other to finally make this dominant allele be realized in your body. So this is the basics of Mendelian genetics. Now, what we have to look at is we have to look at inheritance. So I talked about how you get we have two alleles that code for each of our characteristics, and one is received from our mom and one is received from our dad, right? Then now our question is, so how specifically do we receive these alleles from our mom and our dad? Well, my mom and my dad is, at the end of the day, they're also humans, right? We often forget that, but they're also humans. So my mom and dad also has two alleles that code for their trait. So for example, let's just stick to this hair analogy, okay? Now, by the way, instead of using S for straight hair and C for curly hair, we always just use one alphabet, but we denote the dominant trait as capitalized alphabet and recessive trait as small letter, lowercase alphabet. So in this case, since curly hair is recessive, I'll just, instead of using C, I'll use lowercase s, okay? So let's say my mom had straight dominant, homozygous dominant straight hair, and my dad had heterozygous dominant hair. In that case, my mom would have had two dominant alleles, and my dad would have had one dominant hair and one recessive hair, right? Now, when my mom and my dad each donates or inherits one allele to me, my mom can either donate number 1 allele of hers or number 2 allele of hers. In this case, there's no difference because both alleles that my mom has are dominant straight hairs. Now, when it comes to my dad, on the other hand, he has two options. He can either inherit the first allele, which is dominant straight hair allele, or second allele, which is recessive, so curly hair allele, right? So when it comes to my dad's inheritance, there are two different options. Now, now we have to actually think about combinations, right? We have to go back to pre-calculus and do all the mathy stuff. Now, my mom could always give us large S, right? But my dad could either give me large s or small s. So the probability, the possible combination of alleles I can have is either largest, or largest, smallest. I mean, in either case, I'm going to end up with having straight hair, right? Because even in the second case, I'm going to be heterozygous straight hair, right? Because this big S will override the small s and make me have straight hair, correct? But the genotypes will be different. So in this case, we say, oh, phenotypic ratio is just 1 to 0 because I will always have straight hair. But when we're talking about genotypic ratio, it's going to be 1 to 1, right? 1 for largest, 1 for largest, smallest. If I want to be even more accurate, I can say to 0. This is 0 for smallest, because it is absolutely impossible for me to end up having smallest genotype. Why? Because my mom does not have small s that she can inherit to me. Do we understand this? Now, since this process can be complicated if we have different combinations, we made this thing called Punnett square. Punnett square makes this process of, branching out all the cases simpler. How Punnett square works is you draw a 2x2 grid, and you put your mom's allele here, your dad's allele here, and you just add the two and write in the respective entry, like this. Now, as you can see, we end up having two large S homozygous dominants and two heterozygous dominants, large S, small s. So we can again say that the genotypic ratio is 1 to 1 between homozygous dominants and heterozygous dominants, while the phenotypic ratio is always 1 to 0, because in any of these four cases, I will end up having straight hair. Okay, so this is all about Punnett square. This is what you need to know about Punnett square, okay? So that's pretty much it for Mendelian genetics, but now we have to look at non-Mendelian genetics. Mendelian genetics is a very ideal case, right? Of course, not every part of our traits work solely, strictly based on Mendelian genetics. And there are some exceptional cases. Now, there are four big exceptional cases you really have to know. First is incomplete dominance. Second is codominance. Third is sex-linked traits. And last is, what was last? Last is, multiple alleles. Now, incomplete dominance is simple. It's literally the same as Mendelian genetics, except when you have heterozygous, when you have one largest and smallest, in Mendelian genetics, the large S overrides the smallest, right? But in incomplete dominance, there is no dominance. There's no strict dominance between the two. No allele can override the other. In other words, the two, since none of the two override each other, both of the characteristics will be shown. So in this case, if we're talking about straight hair and curly hair, what's going to happen is, instead of straight hair overriding the curly hair and making me have straight hair, my hair is going to look like half of them is going to be straight and the other half is going to be curly. Now, you might think this is very odd, but this happens from time to time. Of course, not in hairs, but for example, polka dots. Some cows have both black and brown polka dots. That happened because they're heterozygous. The genetic information that code for their polka dots are incomplete dominance, and one of their allele code for black polka dots and the other one code for brown polka dot. So instead of having one overriding another and only having black or brown polka dot, you have both polka dots appearing on the cow's body. Now, then what is codominance? Codominance is similar to incomplete dominance. Everything else is same as Mendelian genetics, except in a heterozygous situation, this time it's codominance. Both of them are dominant, meaning that both traits will mix and blend and will appear on your body. For example, if we were to talk about hairs, instead of having straight hair, or instead of having half straight and half curly, what's going to happen is slightly curly hair will occur on my head. Okay, so this time, instead of both traits being appeared, the two traits will mix, and that sort of median trait will be shown. So if you were to go back to cows, instead of having both brown and black polka dots, we will have brownish black, that middle color polka dot all over the cow's body. Okay, so this is codominance. Now, codominance and incomplete dominance aren't too hard, but sex-linked traits are very hard. So if you're just aiming for a 5 in AP Bio, I advise you to not really study this because you're only going to realistically see 1 or 2 of these questions. But still, I'll explain just in case. Now, sex-linked traits. This time, these traits are the traits that are related to the sex chromosomes of your mom and your dad. So what happens is, instead of just receiving these alleles, you're receiving alleles from your parents' sex chromosomes. So for example, if the trait is linked to only X chromosome, you won't receive any of them from Y chromosome. So in this situation, if we were to draw a Punnett square, you will end up having like something like this. Do you get what I'm talking about? So the difference is, instead of just A's occurring in all of the chromosomes, A's will only occur in X chromosome and not in Y chromosome. Why? Because it's linked to X chromosome. Vice versa could happen. A trait could be only linked to Y chromosome and appear only in Y chromosome. Okay, but in this case, if we were to talk about a situation where your trait is linked to X chromosome, then only in X chromosome you will have this trait. In Y chromosome, you will have nothing there. So you can't really be affected by this Y chromosome. And this Y chromosome can also be passed to you. In one of the two spots you're receiving the alleles, one could be X, the other one could also be X, or it could be Y. If it's a Y, then it's not affecting your trait at all because Y chromosome does not have this trait linked. So the trait will solely be dependent by the X chromosome. But if you are lucky, well, not lucky, if things happen and you receive another X chromosome, then now the two X chromosomes and the alleles linked to them will combine to determine the trait of this characteristic. So that is how sex-linked trait works. Now, honestly, you just have to solve a lot of questions for sex-linked traits to really understand how it works. And again, this is probably one of the hardest part of this unit. So if you really can't wrap your head around this concept, then I would advise you to just skip. You don't have to get a perfect score, right? Now, multiple alleles. Now, for all of the examples above, I was talking about a situation where you have only 2 alleles, right? One dominant and one recessive. But this is not always the case. There might be multiple alleles. So for example, when we're talking about blood types, we have A type, B type, AB type, and O type, right? We have multiple types of alleles possible. So in this situation, we have A allele, we have B allele, and we have O allele. So if you get AA, then you end up having A blood type. If you have AB, you end up having AB blood type. If you get AO, then you also have A blood type, right? So in this situation, instead of just having one dominant and one recessive allele, you're having multiple alleles. So this can also happen, okay? So, and of course, this won't be considered as Mendelian genetics because Mendelian genetics only look at a situation where you have 2 allele, 1 recessive, and 1 dominant. So these are some exceptional cases that you might have to know, but of course, the most fundamental thing that you have to know is the Mendelian genetics, so focus on Mendelian genetics is my advice. Thank you so much for watching, and most importantly, don't forget to have a great day.